All right, so this molecular cloning, it's also called gene cloning. That's why I threw them both in here. All right, so backstory. So 3 years ago, almost to the day, it was definitely this week, my youngest child was diagnosed with type 1 diabetes. She was having some issues. We took her into the doctor, and they're like, cool, you get to go to the ICU now. And I was like, okay, I guess it's not really optional. So what type 1 diabetes is an autoimmune disease where the T cells in the kid's immune system are attacking the cells that produce the protein insulin that attaches to the sugar and allows the glucose to enter the cells. Okay, so for those of us that don't have type 1 diabetes, our bodies naturally do this. Her body can't produce this. So without this, the protein insulin that she now has to inject every time she eats, multiple times a day, uh, she would die. So 100 years ago, being diagnosed with type 1 or type 2 diabetes was a death sentence. But now, because we have this biotechnology, we can treat this. Okay. And so the way that insulin is produced now is through this process of gene cloning or molecular cloning that we're going to go through. All right. So this is one way that we're going to genetically modify organisms. There's other mechanisms, but this is the one that we're talking about for this class. Okay. So what we're going to do is we are going to take the gene that we're interested in let's just use the insulin gene as a case study. Okay. So we'll take that gene that codes for the protein insulin. And what we're going to do is we're going to add those restriction enzymes to cut out just that gene, right? So we don't need all the other genes in humans. We just want the insulin gene. Okay, so we're just going to cut that little bit out. And then we're also going to take this plasmid. Now, if we remember back bajillions of years ago, it feels like, to I think it was chapter 3 when we were talking about cell structure, these little are prokaryotes, right? Bacteria and archaea. They have one circular chromosome in the nucleoid region, but they also have these other small bits of DNA called plasmids. Okay. And these plasmids can be transferred between different individuals of bacteria. What we're going to do is we're going to take that plasmid, that small DNA molecule, we're going to add to the same restriction enzymes. So we're going to get these same little bits of DNA that are sticking out. Okay. And this is going to be the complement of that sequence. And we literally just take that gene that we want, right? That insulin gene, and we just slap it right into that hole and genetically modify the plasmid. All right, so let's look at what we've been talking about using a little paper model, and hopefully it will make more sense. Okay, so here's our model of our plasmid. Okay, it's a small circular DNA molecule that we find in the cytoplasm of prokaryotes. I also have our human gene of interest. In this case, this is the insulin gene, or at least part of it. Let me put it right side up. Okay. And what we need to do is we need to put this human insulin gene and got to connect it with our plasmid. Okay. So in order to do that, we're going to add our restriction enzymes, and our restriction enzymes are going to cut at the site. We get that into focus. Okay. So what I'm going to do is I'm going to model that activity of the restriction enzymes. So I'm going to find my sequence, and I'm going to cut at that sequence of AAGCT. Okay, so I'm just going to mark in pencil where that cut site is so that I make sure to cut exactly the right way. And I'm going to leave some sticky ends. So here's my restriction enzymes. Looks like scissors. Okay, and I'm just cutting along that restriction site that we just marked in pencil. Okay, and so now I have a sticky end, but this enzyme is going to cut at this site as well. All these little nucleotides are going to get broken down and be recycled, which is exactly what I'm going to do with this paper when I'm done. Okay, so now here's our gene of interest with our sticky genes. We can't put it into our plasmid yet because we got to add our restriction enzymes to our plasmid. Conveniently, that is located in bold, so it's easy for me to find. Okay, now I'm going to use my, if you can see those pencil marks, use my restriction enzymes to cut. Oops. Okay, so now on my plasmid, I have the sticky ends. The sticky ends of my plasmid and the sticky ends of my gene of interest are complements. So now I'm going to use this enzyme ligase to join the sugar phosphate backbones between my plasmid and my gene of interest. So I'm taping these idiots together. Okay, now because of the hydrogen bonds, creating a more stable state for this molecule, our other side is going to also join up the complements of those sticky ends. I'm going to have ligase come in, connect those sugar phosphate backbones. Do it on both strains. And now I have my chimera molecule, or I have my original plasmid sequence now with my gene of interest inserted into that. There's a few things genes that need to be present on this plasmid in order for it to be a good candidate to be used in this gene cloning. Okay, so of course we need to have a origin of replication. We need to have a place where our RNA, well, we need to be able to make copies of this. So we need to have DNA polymerase be able to get in there to make copies of it. We also need to have our gene, right? Our gene of interest, the one that we are interested in copying. We need to have that be turned from DNA into RNA so that it can then become the protein that we're trying to make. Okay, so we're going to do that here at this origin of replication. Okay, that's where we're going to have lots of those A's and T's that have only 2 hydrogen bonds between them so that we can open up that DNA and make copies of it. Okay, and of course we have to have those restriction sites in order for our restriction enzymes to work. They have to have that particular sequence where those enzymes are going to cut. And in this case, we saw right that there they are. That's where we plop the gene of interest into. Okay, and then so like those first 2 of those make sense. This other one's a little bit weird, and we have to do some explanation to make it make sense. The third thing that we have to have is an antibiotic resistance gene. And you're like, cool. Okay, why? And the reason is because we want to use antibiotics to kill all the bacteria that did not pick up this plasmid into their cells. And we want only the bacteria that have this plasmid to survive. So what we're going to do is the bacteria that pick up the plasmid, right, that has our human insulin gene, also, bless you, my son, for freezies, you get an antibiotic resistance gene, so you can make a protein that will prevent you from dying when we put you on this Petri plate that's going to kill all the friends that don't have this plasmid. Okay, so again, growing those bacteria on the Petri plates that have the antibiotics mean that any bacteria that did not pick up this plasmid are going to die, and any that do pick up the plasmid are going to survive. All right. So I've isolated the plasmid. I've isolated the foreign DNA. I've cut the plasmid with restriction enzyme. I've cut the foreign DNA with restriction enzymes, and I've put those together. Now the only thing left for me to do is to convince the bacterium that they want this small piece of DNA and that they should take it up into their cells. Okay. And when they do, then bump, ba da bump, da 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 da, that is transformation. All organisms use the same three-letter codons, right? So the start codon being AUG, right? That three-letter code, that's the secret code in the DNA that is going to give us the instructions on how to build the protein. All organisms find that. So we can put a human insulin gene into a bacterium, and that bacterium is going to make human insulin even though it doesn't need it. And it's because everybody uses the same secret code. So this is meaning that we can use E. coli bacteria that are given the plasmids that have human insulin, Taq polymerase, jellyfish proteins that are going to make them glow in the dark, or any other gene that we are interested in having producing that protein, right? So any proteins that we want harvested, we can get E. coli to make that.